थोड़ी मेहनत करनी
Okay, so good afternoon all. Am I audible to all? Am I
Good
audible to all?
afternoon, sir.
Good
Yes,
afternoon,
sir.
sir. You're audible.
Good afternoon. Okay, so let's wait for 3 more minutes. So all will join, then we'll start. Only 3 are remaining, I think. So Kshitiz seems absent today, so we are not going to wait more for Kshitiz. And okay, now we can start. Okay, so before starting, Atoiba is there? Atoiba? 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 Okay, Atoiba, whether you are, so I'm going to mark absent today. Okay, so you are not responding, it means you are inactive, and inactive students can be marked absent. And Atoiba, so one more thing for you, you didn't presented in last class. So for your assignment, for that you are going to be marked 0. So no further chance will be given to you for presenting. Okay, so now we can continue with our lecture. Okay, so some of you presented very nicely, like Nandini and Sahiba and who was the third?
Yukta.
Yeah, Yukta. Okay, so all of you presented very well. So now we are continuing. I will also cover some of the aspect that you already talked about. So just for a revision. So the thing that we are talking here is the module that is our first module, and the subject that we are going to talk is the DNA fingerprinting. So as I mentioned you earlier that here the terms that you are written here, the DNA is your genetic material that you are going to have, and the most in the fingerprints. And in the fingerprint that we all know that fingerprints are specific to each individual. So fingerprints are like that we are having nowadays at the campus when you're entering, you're using biometric, and the biometric you're using your fingerprints, because fingerprints are your identity. So no two individuals are going to have uh, similar fingerprints, or similar maybe, but not same fingerprints. So that's why there the uniqueness is there in the fingerprints, but we use this term fingerprint with this DNA, because the uniqueness is also there in your DNA. And the uniqueness is there that the sequence of DNA that you're going to have, no two individual is going to have same DNA sequence. So whether we are talking about we get all the genetic material from our parents, but even we are get genetic material from two parents, so we have the mixed blend of that sequence. So none of us our genetic sequence is matching 100% to our parents. Simple, you can say it's definitely going to match, but it's not same. So that's why we can say that the individuality is there in this DNA sequence. And when you reference the DNA simply means it's a chain of nucleotide, and what pattern your ATGC are patterned, so that we is going to be. So this, as I'm talking about to you, this is, as you can say, over the right you are saying your thumbprint, and over the left different panels are there. In this panel different bands are there, and each band is representing your DNA sequence, and DNA profile is going to be there, and each band is referred to the size, and this size is going to be the pattern that you're going to generate. That is going to depend upon what marker you are going to use. So before continuing the things, let me just explain you what are the markers and okay, sorry, I got disconnected in between. Sorry, students. So there was an issue in the network, so that's why it is connecting and disconnecting. Sorry for that. Okay, so I was talking about something that, as I mentioned, you. I asked you in your previous semester. I think have you heard about the markers? Markers.
Yes, sir.
So can anybody define the term markers? To what we are talking, to whom we are telling the markers?
Sir, can I?
Yes.
So, markers are basically can be short genetic sequences which are, in a way complementary to a particular sequence that we are searching for. That is a sequence of interest, and when it is added to, let's say, any of these DNA profiles, then it will go and identify the sequence that we are looking for. So, it tags or marks the sequence of interest, which helps us identify it.
Very well said, Manaswini. As Manaswini told that these markers are the sequences. These are the that are present on your genome, which you can mark. Simply, just I'm using this term marker. The sequence which you can mark. So they are the sequences, or though they can be recognized, or whose presence and absence you can record. So those you can tell that they are the markers. So in your previous semester, you heard about two, three things. If you remember, one thing you studied, that is the your restriction endonucleases. So your restriction endonucleases have one specificity that they can recognize a specific sequence. If that sequence is going to present in your genome, then it is going to be digested. If that sequence is not present, then it's not going to be digested. So I mean that presence and absence of that sequence in for that restriction endonuclease can tell you that the marker is there or not. So restriction sequences also can serve you as a marker. And second thing is that in case of your PCR, PCR you utilize the primers. So primers also can you use as a marker whether that sequence is going to present in your genome or not. Okay, so so one thing you note that it is a bit different from DNA sequencing. This DNA fingerprinting bit different from the DNA sequencing. Both are different techniques. In DNA fingerprinting, we do not talk about the whole genome sequence. We just talk about the markers. Okay, so coming back to just as we are at the introductory one, so in detail we will go a bit later. So just by definition, if you define what DNA fingerprinting is, so DNA fingerprinting is just a chemical test that show the genetic makeup of a person, of or other living things. So what you are talking about? You are talking about the genetic makeup. You are not talking about the genetic sequence, whole genome sequence. We are not talking about. We are just talking about genetic makeup. So what it is purpose? So it is used as a evidence in court. So legality is there. So legal issues are there in it. You can identify your bodies. Like if you remember a few days back, our CDS General Vipin Rawat, who died in a air crash, in a helicopter crash, and so all the bodies were so much burned that you cannot identify just by looking them. So for identification of them, this DNA fingerprinting were being used. Using this DNA fingerprint, their bodies were identified. Track down the blood relatives, and to look cures for a disease. So these are the few things for which you are going to utilize DNA fingerprinting. But by definition, what it is, how to show, to check genetic makeup of a person. And another way you can also define it way that DNA fingerprinting is a laboratory technique used to establish link between biological evidence and suspect in a criminal investigation. So simple thing you can note that this DNA fingerprinting we are talking about, this DNA fingerprinting is a technique which is going to utilize for forensic purpose. So the main purpose of this DNA fingerprinting is going to be in the forensic, for the identification, for the connecting links. So there is going to be a DNA fingerprinting. So when I'm talking about anything, we always used to talk about its history. So how it is going to be came into the discovery, how we are talking about this is a DNA fingerprinting. So historical background, if you simply all of you are young students, so you like novels or some good some things that is like Sherlock Holmes. So everyone would like to like detective agents etc. So if you just go back in the Sherlock Holmes story, what the Sherlock Holmes used to use some terms or some words like that. I have found it. I have found it. He shouted, and running toward us with a test tube in his hand. I have found a reagent which is participated by hemoglobin and by nothing else. Say Sherlock. So this is his novel words or his like his serial things. And she says Sherlock Holmes to Watson and Arthur Conan Doyle, a first novel, a study in Scarlet from 1886 and later. So now whatever that Sherlock was talking about, he found something. He found a reagent that from the blood something, so that we later we relate this what we are having nowadays. So now we have Sherlock Holmes test, and there is will no longer be identified. So this is a Sherlock Holmes story that Sherlock Holmes described that now I'm having a test. By using my test, you can tell who is going to be the criminal. So but now recent time we are a scientist, and this really that Sherlock Holmes test that we are referring, that Sherlock Holmes referring in 1886 was discovered around 100 years later in 1984. So this is a name that scientist that you are going to see here. The name of the scientist is going to be Alex Jeffrey. So he's the one who finds the real Sherlock Holmes test. So earlier in the novel Sherlock Holmes, whatever the test was written there, no such type of test was there. But the real test that was discovered, the real Sherlock Holmes is Alex Jeffrey, and he is also called as a father of DNA fingerprinting. So what this Alex Jeffrey did and what he was doing, so the Eureka shout shocked England again and was heard around the world when it is roughly 100 years later, Alex Jeffrey Where he was in the University of Leicester, in the United Kingdom. What he was doing, he was studying the myoglobin. You can just combine what the Sherlock Holmes referred. He was referring hemoglobin, and what Jeffrey was studying, he was studying the myoglobin. So myoglobin and hemoglobin both are going to be the part of your blood proteins. So what he found, what he was doing, he was studying gene. He was not talking about this protein itself, but he was talking about a gene which is going to code for this myoglobin. And what he found the extraordinary variable and heritable pattern. So what his discovery, right? There are the variability. So what we are going to talk about in DNA fingerprinting, we are and whatever the thing when we refer, like if you talk about, if you two individuals are there, if you see two individual, you recognize them separately. Why we are recognizing them separately? Can anybody comment? Why we recognize two individuals separately? Just a minute, I'm coming. बोलो। मुझे खाना बना दे आपका। क्लास के बाद। Sorry, student. Okay, so my question, simple question, if to you, how we could recognize each other? Like Manaswini was talking about. So I recognize that she is Manaswini, even she didn't turn on the camera. And but when the camera is turned on, then by seeing each other's face, we can recognize each other. Why? How we are going to be able to recognize each other's? I'm audible to you?
Sir, because we have characteristics and features which are identifiable in each person.
Simple
So
thing
because we
in
have memory.
no, that's a even without memory we can differentiate two are the different. Okay, so simple thing is the variability. Everyone are varying, differing with each other. But you simply talk about that every one of us are going to have more similar feature rather than the dissimilar features. Like every one of have two eyes, one nose, two lips, two ears, a forehead, hairs, chin, two hands. Everything is, most of things are similar, but few things are which are not similar. But so simply you can say that what we are focusing, we are focusing on the variabilities, what things are differing. Even a minute difference is there. So like simply you say that the stiff, like you are mostly you are girls, and whenever you go to a shopping for shopping, you can differentiate two different colors. Like girls are able to differentiate even very minute differences. The thing is that our focus is always on the differences, not on the similarities. So only on the basis of this dissimilarities we distinguish. We find two are the different. We are not focusing on the what similar is there, but what we are focusing on the what is not similar. Okay, here also this Alex Jeffrey, when he was studying the gene sequence or sequence of gene that is called in the myoglobin, he found that there is extraordinary variability compared to other genes. This myoglobin genes have much more variability. Like if you simply you can say that two individuals do not have a same sequence of this myoglobin. Maybe like amylase gene that I am having, you also have the similar sequence of the amylase gene, but certain genes are there which are having extraordinary variable things. When the variability is there, you can tell that these two are different. If variability is not there, then you can't say that these two are different. Like simply I give the example of our CDS Vipin Rawat. All and the whole, In that chopper there were 10 individuals, and those 10 individuals, they are burned. And after burn, when they got burned, you are not able to distinguish them, because the dissimilarity, what the variability that we are talking about, that our eyes can distinguish, that is just simply their faces. On the facial variability we can recognize. If facial variability is not there, then our eyes cannot recognize. So that is the thing. So when so we are going to focus on which thing you can recognize. So so simply things, the variability, if variability is there, then you can recognize. If variability is not there, then you can't recognize. So we have to find certain things. So what this the Electrophoresis did, in after 100 years that Sherlock test, he find that variability, and that variability was there in this myoglobin, the gene that he found. And one more thing was with this gene was that it is heritable also. Like when the variability is there, it's not variability, it doesn't mean like if I got a variable sequence in me, then this variable sequence is just staying with me, it's not heritable. Yes, this variable sequence is further moving to my offsprings also. Okay, so that is his discovery. And what form it was? It was in the form of repetitive DNA sequences. And what he used the repetitive DNA analyze and multiple DNA focus probes he's going to utilize. And not being Holmes, he refrained to call a method after himself. Like Sherlock Holmes used the term Holmes test, Sherlock Holmes test, but Alec Jeffrey do not call it as a Jeffrey test, but he used the term DNA fingerprinting, because we scientists doesn't believe in our own name. We believe in the techniques, the technology, what it is going to be. So continuing the history, if you focus, so where it is going to be utilized, the process developed by Jeffrey in conjunction with the Peter Gill and Dave Watts of the Forensic Science Services. So still continuing the history, where you going to utilize it, where the study of how this DNA fingerprinting, that Jeffrey's work came into the picture. So the Jeffrey, as I mentioned, that Jeffrey was working on around 1984. It just doesn't mean that he started working even in 1984 only. He was working few years back. So what happened while he was working, this first application of his techniques came during that time itself. So what is the utility of his DNA fingerprinting? What it did, it does not save a young boy by convected. His technique, he saved a young boy at a time in 1984, and he also make that real criminal got convicted. So this is what his story was. His story is associated with the two teenage girl. And the name of these teenage girl was Linda Mann and Dan Ashworth. What he was there, who were raped and murdered in 1983, and in the where they were got in the English village. So, and at similar time, things also occur in 1986. So now at that time there was no Sherlock Holmes test, but what happened during that time, as I mentioned, they were raped and murdered. So from that crime scene, the semen sample was obtained. So now, after discovery, these the inquiry were made. Detective David Becker were hired for them, and this Detective Baker, what he did, he contacted the Alec Jeffrey, and then they talked. They found that then Alec Jeffrey mentioned that yes, using my technique, we can try to solve this case. Then what they did, they collected DNA sample from the blood and blood of all the villagers. So like this village, at that time, the population of village is around 5,000 people were there during that location. So what they did, they collected the blood sample of all volunteers from around 5,000 local men who willingly assisted and assisted the investigation. And what happened at that time, the real the what the who we can say the real criminal was misjudged. So what he did, The Richard Buckland, an initial suspect who had actually confessed to one of the crimes, and subsequent conviction of the Colin Pitfalk in January 2, 1998. So what he did at that time, he initially he sent, instead of him, his maid, his worker for the blood sampling. So at that time he got escaped. But so as I mentioned, the one of criminal was caught. Then recollection were made, and the recollection leads to the final final catching of the real criminal. So just simply, if you just we read out what really happened at a time. Pitchfork, a worker at a local bakery, had course his work colleague Ian Kelly to stand for him when providing a blood sample. So Pitchfork, instead of him, he asked his work colleague Ian Kelly to provide a blood sample when they used to forge the passport and impersonate Pitchfork. Okay, so this is the thing how happened at a time. The deception was discovered and subsequently reported to the police after Kelly was overheard boasting of deception in a local pub. Police first arrested Kelly and then Pitchfork, whose blood was sent to Jeffrey's lab for processing and profile development. Pitchfork profile matched the DNA left by the murderer, which confirmed that Pitchfork presence at both the murder scene. Pitchfork then confessed to committing the crime after he was conferred with the evidence that his DNA profile matched the trace DNA from two crime scene. So this is the crime scene that happened in 1983 and 1986, and utilizing the technique that developed by Alex Jeffrey, and that leads to the conviction of Colin Pitcock in 1984. So even at the time of discovery, when this Alex Jeffrey discovered this technique, it was utilized, and it helped in conviction of real criminal at that time. Okay, so this is a simple historical evidence and historical example how DNA fingerprinting being discovered and utilized for. Uh, just a minute. Uh, Okay, so this is initial story of DNA fingerprinting. Then subsequently, it was utilized within a 2 year of time. This technique were uh, advertised and utilized worldwide. But it was around 1987 when companies such as Cellmark and Academic Medical Legal Institute Around the world, national police, law enforcement agency, and so on started to evaluate, improve upon the employing new tools. The year after the discovery of DNA fingerprinting were characterized by a mood of corporation and interdisciplinary research. So now this just one news, one story, one successful case resolving leads to the utilization of this thing worldwide and more research, because as I mentioned you that this differentiating and distinguishing two individuals is all based upon the differences. And so you need differences are going to be the based on the markers, that term that I use, the marker, that like you can mark the location. So you need a new marker. You can't just rely on just one marker. No, you can't just rely on just myoglobin. So discovery researchers keep on advancing, and new markers were being discovered and being now are being used. So DNA fingerprinting Congress held on the 5 continents in Bern 1990, in Belo Horizonte in 1992, even in India, Hyderabad in 1994, Melbourne 1996, uh, Peter Elizabeth 1999. And this is when you are saying that this Congress is being held. So this Congress simply means that so people worldwide who are working on this field of DNA fingerprinting, they came together, they sit together, and they design and they decide a mandate, but how we can proceed to further strengthen these techniques. Because when we are saying any techniques, even in biological techniques, when you're talking about fingerprints also, you can force them. So where are you finding that the chances of forging and chances of because here the and this technique you're going to utilize in a forensic, so always the life of a person is on the stake. So that's why with a good feeling that job was done. So the period in 1990 was the golden research age of the DNA fingerprinting, succeeded by 2 decades of the engineering. So this is the time when the researchers were on. And just a moment. हाँ जी डॉक्टर साहब, हाँ जी ना क्लास में हो रही तो अच्छा जी, तेरे पास आया था क्या? बना रहा हूँ, बनाता हूँ, बना ली, क्या करना है उसमें? हम्म। Sorry, student. So I got a call from the university. Okay, so simply just what if I talking about? I just talking about the development of this field and how we got to the current position. Main research were happened in the 1990s, means early, and that was a time of the DNA fingerprinting. So every research, even at the time when I was in college or in the early master's degree, like you are the undergrad and after that. So at that time, whatever the research in my university was going on, all the research was on the DNA fingerprinting. And the DNA fingerprinting not just focus on the as aspect of human, but also the aspect of the plants. So because the main thing, the aim of this DNA fingerprinting is identification. So using it, you can identify different traits like simply if you remember that if you select a plant, for example the rice, so rice need lots of water. So if you found a rice which can even grow in a low water requirement, so what are the genes that are present in that rice that are going to that are responsible for growth in low water requirement? So all these things you can identify using this DNA fingerprint technique. So Jeffrey's original technology now obsolete for the forensic use because the technique was based on the only one gene and underwent the importance of the development in term of the basic metrology, that is from the southern blot to the PCR. So whatever the technique that Jeffrey referred, that is Jeffrey was based on the, because at that time PCR was not there. We all relying on this restriction nuclease or the P probes, southern blotting. So that is a bit time consuming things. So now we move from the southern blot to the PCR, and radioactive probes were being utilized at a time, and now we are using not radioactive probes. So now we can use fluorescent probes and going to be a bit less toxic. And slab gel electrophoresis. Now we are having capillary gel electrophoresis, and the capillary electrophoresis, even saying one time you can run large number of sample, and the length of run is also going to be much more. Like in slab gel, you know that just a 10 centimeter, 20 centimeter gel you can run, but in capillary electrophoresis you can run in meters because capillary is going to be very thin, and you can roll its capillary also in a circular, in a round shape. Large capillary you can accommodate in a very small volume or a small area. So that's why and that's why DNA fingerprinting was termed as a DNA profiling, because you are generating a profile of DNA. You're not generating a fingerprint. Fingerprint is entirely a bit different thing, but both DNA fingerprinting and DNA profiling profile you can use for the same thing. But what counts the bit for case and still counts today is the process to get DNA identification result accepted for legal proceeding. Spectacular flashes from historical 1989 case of people versus Castro, New York, when the case against Knox and Solicitor in Italy. So these are the newer recently, not just Alex Jeffrey case, but also this recent case were resolved using the DNA fingerprinting. And but these case also discovered, they found that some severe insufficiencies are also there. So, means when you're saying that these two individuals they are same, but their margin of error is very little. So you need to further go for a technical advancements, technical protocol, especially DNA evidence, and while you're interpreting it, and so you must have to eliminate all the doubts which are there. Then only you can use this DNA fingerprinting as an legally acceptable evidence. Okay. So now I think I should like to conclude because time is also running out, and I also joined to a meeting. So please turn on your camera for recording your attendance. Uh, Adesh, Adesh, Adesh,
Present, sir. Present, sir.
turn on your camera also. Adesh, turn on your camera.
Yes, sir. Sir.
Atoiba, Ayushi,
Present, sir.
Demetrius,
Present, sir.
Devanshi,
Present, sir.
Ishita,
Present, sir.
Janvi,
Present, sir.
Kshitij.
Sir.
Kshitij, why you are joining late? You made it a regular practice, so you will join after 15-20 minutes.
सर अगले बार मैं फिर ठीक टाइम में join.
So this time you will be marked absent. So next time you will become regular, then I will mark you present. Because I have given you attendance from last 3-4 times. Manaswini, Dan, Namrita, Nandini, Namrita,
Present, sir. Present, sir.
Nandini,
Present, sir.
Nehal,
Present, sir.
Rupal,
Present, sir.
Sahiba,
Present, sir.
Sanjana, Shakti,
Present, sir.
Shivani,
Present, sir.
Tanvi,
Present, sir.
Tridiv,
Present, sir.
Vivek, Yukta, Yuvraj.
Present, sir.
So thank you all. Stay safe, stay happy. See you again
Sir, I'm
in our next
present.
session. हाँ विवेक हो तुम भाई हो.
Thank you, sir.
Welcome.
Sir, excuse me.
हाँ जी बोलो.
सर आपको पीपीटीज के टॉपिक्स बताने थे आज.
हाँ मैं अभी बताता हूँ आपको. मैं आपको इसको देवांशी को बता दूंगा.
ओके सर.